Hi everyone, I'm Kalaimani Vetrivel from SNS College of Pharmacy and Health Science. So today we see about glycolysis. Glycolysis is nothing but the process in which glucose is broken down to produce energy, and it produces two molecules of pyruvate, ATP, NADH and water. And this process takes place in the cytoplasm of a cell. In mitochondria, that is cytoplasm of a cell, and does not require oxygen. It occurs in both aerobic and anaerobic actions. See, a first step is a phosphate group is added to the glucose in the cell cytoplasm, and by the action of enzyme hexokinase. In this, a phosphate group is transferred from ATP to glucose, forming glucose 6 phosphate, and glucose 6 phosphate is isomerized into fructose 6 phosphate by the enzyme phosphoglucosamidase. Other ATP molecule transfers a phosphate group to fructose 6 phosphate, and it converts it into fructose 1,6 biphosphate by the action of the enzyme phosphofructokinase. And the enzyme aldolase converts fructose 1,6 biphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which are isomers of each other. Triose phosphate isomerase converts dihydroxyacetone phosphate into glyceraldehyde, and in the successive step of glycolysis. And here one ADP is also converted, that is excreta. And now you can see the glucose 6 phosphate isomerized. It's an open chain, and enzyme formed. Glucose 6 phosphate is getting isomerized and it's converting itself into fructose 6 phosphate. And here phosphoglucomutase enzyme is being activated. This is the step 2. A ring form of fructose 6 phosphate have been obtained, and from this 1 ATP molecules have been generated, and from fructose 6 phosphate converts it into fructose 1,6 biphosphate. And here phosphofructokinase enzyme is acting 1,6, a ring form of fructose 1,6 biphosphate have been transferred. And now here two molecules of ATP and two molecules of ADP have been generated. And in the step 4, see, the enzyme aldolase will help fructose 1,6 biphosphate into the conversion of glyceraldehyde, and that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which are isomers of each other. That is glyceraldehyde 3-phosphate is the isomer of dihydroxyacetone phosphate. Here the enzyme which is acting as aldolase. So you can see how it is getting isomerized, and dihydroxyacetone phosphate is have been obtained. Glyceraldehyde 3-phosphate is also been obtained. And here, after the conversion of glyceraldehyde 3-phosphate and say if it is dihydroxyacetone phosphate have been isomerized as a glyceraldehyde 3-phosphate itself. Now you can see how they are both are the isomers. And in step 5, from glyceraldehyde 3-phosphate triose phosphate isomerase will convert this dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, which is the substrate in the successive glycolysis. This step undergoes two reactions. This is the step 6. The enzyme glyceraldehyde 3-phosphate and dehydrogenase transfers one hydrogen molecule from glyceraldehyde phosphate to nicotinamide adenine dinucleotide form NADH plus H plus. And glyceraldehyde 3 phosphate dehydrogenates add a phosphate to the oxidized glyceraldehyde phosphate to form 1, 3 by bis phosphoglycerate. Then phosphate is transferred from 1, 3 bis phosphoglycerate to ADP and to form ATP with the help of phosphoglycerokinase. Thus, two molecules of phosphoglycerate and ATP are obtained at the end of this reaction. And in step 8, the phosphate of both the phosphoglycerate molecules is relocated from the third to the second carbon to yield two molecule of 2-phosphoglycerate by the enzyme phosphoglyceromutase. The enzyme enolase removes a water from the molecule 2-phosphoglycerate to form phosphoenol pyruvate. See, the water molecules have been removed and you obtain phosphoenolpyruvate. And a phosphate from phosphoenol pyruvate is transferred to ADP to form a pyruvate and ATP by the action of pyruvate kinase. And two molecules of pyruvate and ATP are obtained here. This as the end product, that is two molecules of pyruvate and two molecules of ATP. So net energy is 280